Hello viewers, welcome back to my channel. Today's video we're gonna discuss about the ribosomes in the protein synthesis, chemical composition, mechanism and etc. So let us start. Before that, please do share, like and subscribe to my channel. Stay tuned. Thank you. Let us start today's session. Start with the protein synthesis in ribosomes. Ribosome synthesized from nucleolus attached to the mRNA. Here there is a termination end. They get relieved and those relieving mRNA synthesize protein. And when it required to the cell, it comes out from the cell depending on the requirement. If it is not required, it is not gonna come out of cell. Here what happens, the ribosomes are synthesized from the nucleolus that is attached to the mRNA. Here the mRNA has a termination end. From that the mRNA gonna relieved or released. The relieved mRNA is gonna synthesize the protein. Here mRNA comes from DNA present in nucleus. Eukaryotic ribosomes are more weighted than the prokaryote. When centrifugate, eukaryotic ribosome stays lower whereas prokaryotic ribosome settle above. And then come to the chemical composition. Here during the sedimenting the smaller and the larger unit. Here in ribosomes, as I said before, two units are present. The larger unit is dome shaped and smaller unit is the base or plate shaped. So when sedimenting these two units, the smaller unit settles at the top as its molecular weight is low as compared to the larger unit. So 50s and 30s is the Swedberg unit when the two units are in detached condition. Here the whole Swedberg unit of the prokaryotes is 70s. But when these are in the condition of detached, not attached, when these are in the condition of detached or separate, the Swedberg unit is the larger one is 50s and the smaller or the base is 30s. And then in prokaryotes, three different types of RNA can be observed. 5s RNA, 28s RNA and the 16s RNA. Here the 28 and the 5s RNA are present in the larger unit and in smaller unit 60s RNA is observed. How we know about this? When we extract the animal tissue or the fluid, when we consider that for the centrifugate, the RNAs of different molecular weight gonna settle at the different positions. So the 5s stays at the topper position as the molecular weight is low, and then the 28 is gonna settle at the below fully bottom, and the 16s is settled on the middle of the 5s and the 2s. So depending on the molecular weight, we are going to name this as a 5s, 28s or 16s. And then in eukaryotes, there are four different types of RNA can be observed. Firstly, 5s RNA, 5.8s RNA, 28s RNA are present in the larger unit, and then the 18s RNA is present in the smaller unit. And then in prokaryotes, 60% RNA is present and 40% proteins are present. But in eukaryotes, 40% RNA is present, 16% protein is present. Why this difference? prokaryotes, RNA is more important as bacteria needs the RNA, but in eukaryotes, the plants, fungi, etc. are need of the protein, not much RNA. So on the requirement it is differentiated, and then the protein is more because it is complex. In eukaryotic cell is the complex cell, hence the protein is more in number or in ratio. Some parts of lipids and metallic ions also present like Mg2 plus, Ca2 plus, calcium and magnesium are present. Here the composition may varies, but it reaches only till 80%. In different cells the composition may vary, but it till only reaches 80%. 50 to 55 proteins are observed in prokaryotes, in large 34, in small 21, along with RNA. Hence these are known as the protein factories. And then come to the mechanism. During initial stages, two units are attached, not attached, it is detached. Then when the concentration of magnesium and calcium increases, it gets attached in nucleus. DNA converted into the mRNA. mRNA is known as the messenger RNA. Here the mRNA attaches in between two subunits. In larger unit, tRNA transfers RNA, it functioning in the searching amino acids. tRNA function is the searching of the amino acids. Where from the amino acids are came? Amino acids comes from the food what we intake. And then it sends amino acid to another tRNA, and that receive particular tRNA comes out and form chain like structures, and all these amino acids becomes protein. Here consider a cell having or a ribosome having a 2 tRNA. Here one tRNA is receiving the amino acids and mRNA, and it sends it to the another tRNA. Here another tRNA receiving, and the particular tRNA comes out the amino acids and forms a chain like structures which gonna synthesize the proteins. So thank you for watching. Please do share, like and subscribe to my channel. Stay tuned. Thank you.